While technology more, very feasible to us through be the updated versions of iOS, Android, or Play Store updates, keeping up with this scientific advancement is almost impossible. So, in an humble attempt to keep you all updated with the latest developments in the field of chemical sciences, we have arranged this seminar. We have the privilege of hosting two distinguished international speakers who will be sharing their expertise and insights in their respective fields. On the first day of our seminar, we have the pleasure of welcoming Dr. Naresh Sanandia, who will be discussing the topic of modification of natural polymers for their biomedical applications. And on the second day, we will be joined by Dr. Masashi Su, who will be presenting on the topic of radical reactions on interstellar icy displays. Some common instructions before we begin to all participants is that after each day presentation, we will have a Q&A session where attendees can ask our speakers questions and engage in discussion on the topics of presentation. So we highly encourage active participation from all the attendees to make the most of these valuable opportunities. And we will also be providing an attendance link at the end of each day to record your participation and receive any feedback that you may have. Now, I would like to invite Dr. Keyur Bhatt, Head Department of Chemistry, to share his opening remarks and motivate our students to take full advantage of this opportunity. But, sir, please.
Yes. Very good afternoon to all. Am I audible?
Yes, sir, you are audible.
Okay. So, good afternoon, Dr. Naresh.
Hi, good afternoon.
Hope you can spare time for your schedule. So, I welcome once again to all participants and faculty members. I invite you and welcome you to today International Desktop Seminar on Chemical Sciences. For this, we have two eminent speakers from UK and Japan. Dr. Naresh is from Research Institute, School of Engineering and Newcastle University at UK, and Dr. Manashi is professor from Institute of Low Temperature Science, Hokkaido University, Japan. So this two-day seminar we arrange for the students, those who are interested in the particular chemical sciences, natural polymers, radical reactions, and some advanced topic of chemistry. So hope you all are enjoy this 2 days desktop seminar. We have some guideline and instruction for this participant. We'll share you during this webinar session. So without wasting of time, I just hand over my mic to Jamie to introduce our speaker. Thank you everyone.
Thank you, Bhatt sir, for your valuable insight. We will now proceed with our first presentation of seminar, and I would like to invite Dr. Hasit Wagani, Assistant Professor, Department of Chemistry, to formally introduce our esteemed speaker for today. Over to you.
Yes. Am I audible?
Yes, sir, you are.
Yes. Thank you, Jaimin. Dr. Naresh Sanandia is currently a research associate in the School of Engineering at Newcastle University in United Kingdom. He has carried out his PhD work in CSR, Central Salt and Marine Chemicals Research Institute, CSMCRI at Bhavnagar, and awarded a PhD degree in chemistry affiliated at MK Bhavnagar University. Following this, he has pursued postdoctoral research at Pohang University of Science and Technology in South Korea, and later at Singapore University of Technology and Design in Singapore. He has published 25 plus articles in renowned journals, publishers such as Nature, ACS, American Chemical Society, RSC, Royal Society of Chemistry, Wiley, Springers, elsewhere, etc. And also has 30 plus media coverage in various national and international media houses. Additionally, he has delivered talk at various national, international conferences and universities. So with this, now I invite Dr. Naresh to deliver his talk.
Okay, let me share the screen.
Yes, your screen is visible now.
Okay, good afternoon, everyone. Good afternoon, everyone. Thank you for joining this talk today. And I would like to thank Dr. Hasit for kind words and also organizing team for this platform. So I'm very much delighted to be here today to share my knowledge and expertise on the topic of natural polymers for biomedical application. I have been passionate about exploring the potential of natural polymers as a viable alternative to synthetic one for the biomedical applications. My research has focused on chemical modification of the natural polymer to harness unique properties to create innovative materials that can be used in various a variety of medical applications. So I'm excited to share with you some of my research in this area, and I hope that this talk will inspire you as it has inspired me throughout my career. So, okay, can you see my screen? Yeah. Okay, I think you can see my screen and you can hear me. So this is the outline of today's talk. So I will start with little bit from the basic, because many of you might not aware about like what is the natural polymers or biopolymers, and like what properties they have. So the outline of this talk will be biopolymers, like introduction of the biopolymers, and then what are the opportunities and challenges while we use these polymers for biomedical applications, and then functionalization of biopolymers. So like how we need to change the polymers to like harness like a better properties or a desired properties for especially for biomedical applications. So those were the introduction part, and then I will also share my research projects that I have carried out on this topic basically. So those are developing of surgical glue or a tissue adhesive that's used in a surgical process, and then a development of guided bond regenerative membranes, so that is for dental applications. And then another one is dentinal hypersensitivity treatment solutions that we have developed, and then injectable hydrogels and 3D printing. So yeah, you can ask, or you can raise a hand if you have any question from these slides, or you can ask at the end of presentation. Um, yeah, so yeah, as I said before moving to my research, I would like to give a brief overview of fundamental concept of how biopolymers works, and I mean, for those who are not familiar with them. So biopolymers or natural polymers are large molecules found by linking smaller units known as monomers. It's same like synthetic polymers, but synthetic polymers we develop in a lab. So we do like chemical reactions, and we just chemically attach the monomers together, and that forms synthetic polymers. But in biopolymers, it occurs in a nature. So we basically found ready-made. Of course, we need some purification, but yeah, those are from biological sources. Um, and they are derived from living organism as well, and is readily available from renewable resources. Biopolymers can be or natural polymers, basically those two are almost like a similar term, biopolymers or natural polymers. So it can be classified into like several types based on their chemical structures, origins and functions. So here is a common classification of a natural polymer or a biopolymer. So it is divided into major three categories: nucleic acid, proteins, and polysaccharides. So nucleic acid are basically polymer of nucleotides as a building blocks. And then the examples of nucleic acids are DNA and RNA. The another natural polymer is protein. I think we all know that. So it consists of amino acids, and the examples are like hemoglobin, collagen, trypsin, all enzymes, etc. And then the last one is polysaccharides. So those are basically made of glucose or similar simple sugars combining and form a polymer. So those are the classification. So in polysaccharide, there are like a starch, cellulose, chitin, agarose, carrageenan, those kind of materials. So here in this talk, I will mainly focus on cellulose and chitin. These two polysaccharides. So basically, it's about the polysaccharide modification. So yeah, cellulose and chitin. So these two are let's talk about cellulose. So cellulose is a polymer composed of glucose unit and is linked with beta 1,4 side, and that forms polymer. And it is the most abundant organic compound on the earth. And basically, cellulose is a structural polysaccharide in plants. So in all trees or like all trees or vegetation, it consists cellulose. While on the another hand, chitin. So chitin is the second most abundant biopolymer, and it is basically structural polysaccharides in animal. Structural polysaccharide means like these small animals or plant, we have a bond in our body, but they don't have bond, right? So these cellulose and chitin act as a support for their structure. Um, so yeah, chitin is composed of N-acetyl glucosamine unit, and it's a repeating unit in the chitin. So if you see the structure of chitin, you can see like an acetyl is NHCOCH3 group, right? So that is chitin. But if you deacetylate the group, so instead of NHCOCH3, if it is NH2 only, then it makes chitosan. So the chitosan is a derivative of chitin, is basically a deacetylated chitin. So yeah. So polysaccharides is have like a very much advantages in the biomedical application because of its properties. So it is biocompatible, biodegradable, pH sensitive, mucoadhesive, antibacterial properties. It has a non-toxicity because it is from biological sources. Um, so which makes them ideal for use in biomedical application. So biocompatibility refers to the ability of a substance to evoke no adverse response when it in contact in biological tissues, while biodegradability means is ability of material to break down into simpler substances when in contact with biological tissues or biological surroundings. So these are the applications that biopolymer is used mostly. So his the common structure of biopolymers is mainly sorry, polysaccharides is as I mentioned, it's a simple sugar, and it has a different functional groups. So that is basically all the polysaccharides have different physicochemical properties. It is used in these like various type of applications. So yeah, so let's have a look at the opportunity that these natural polymers or polysaccharides offer. They are renewable, which enables us to reduce our reliance on substance on sustainability, and in addition, they are sustainable in terms of their biodegradability, making them environmental friendly as they break down naturally over the time. So it's a sustainable, renewable, and also is, as I said, is abundant, like most abundant and second most abundant. So um, they are very cost competitive. It's very cheap and it's everywhere. Biodegradable, that I already mentioned, non-toxic, it doesn't generate any immunogenic effect in the body. So most of them are non-immunogenic, shows non-immunogenic behavior. Um, so despite these properties of the polysaccharides, these polysaccharides do have some challenges that must be strategically addressed to ensure their successful use. One of the biggest impediment is their undesired physicochemical properties. Um, yeah, is sometimes it's not suitable for tissue engineering application. And another one is insolubility in most solvents. So for example, cellulose and chitins. So those two polysaccharides are mostly insoluble in most solvents, like unless you use harsh solvents. They have very limited functional groups. It's not so much active, like I mean, it's like an acetyl, as I showed, and hydroxyl group. So it's not active functional groups. It has a low surface area, poor mechanical properties, and it shows uncontrolled swelling behavior when it transforms into the hydrogel. So these are the challenges while we use polysaccharides for any biomedical application. So that's why we need to get rid of those challenges, or to get the desired properties, or physicochemical properties. We need to change the chemical structures of these polysaccharides. So there are different ways to change the chemical structure. So for example, let's talk about the chemical modification. So in chemical modification, we change the functional group of these polysaccharides. So basically, when we have a hydroxyl group in cellulose, we convert it to the either carboxylic group, or methoxysilane group, or sulfur containing group, esterification, oxidation, phosphorylation. So those kind of strategies are used to alter its physicochemical properties, basically. And another type of modification is physical modification, which means either we use blending with another natural polymers or synthetic polymers, or we do cross-linking with with different cross-linkers. So those are the physical modification, and there are some fabrication techniques as well. I mean, to modify at a very small scale. So it's basically nanofibrillation, and then yeah, it's like composite formation. Then we get hydrogels, nanofibers. So those kind of the functionalization are used for the to get the desired properties of these polysaccharides. So these were the overview of what are the biopolymers, and then what is polysaccharides and cellulose and chitin. So let's talk briefly about some of my research projects. So this project is on tissue adhesive. So tissue adhesives are known to accomplish a diverse function for internal tissues during hemorrhage or surgical procedure. So when we have like a surgery in our body, when there is bleeding, like internal bleeding during the surgery, so we need to stop that. So it's like we need to stop that. So there is this glue that is used normally, and yeah, there are like many commercially available glues as well. So that's the tissue adhesive. So this is different than our like normal paper glue or something, because in our body the environment is wet, right? It's all water. So these adhesives should be work in a wet condition. So that's why it is different than normal adhesives. And also choosing material, because we are like putting in into our body, right? So it shouldn't be toxic, as well as it shouldn't make substances that are toxic to our body while it has like degradation. So that's why the tissue adhesives are unique on their own. So we have developed these chitosan based wet tissue adhesives with hemostasis function. So hemostasis function is basically a material to stop the blood flow. So that is the hemostasis. So we have developed this tissue adhesive with hemostasis function inspired from nature strategy. So that is biomimicry. So we have used biomimicry, means we copied a mechanism from a nature. So if you see here, it's I don't know if you can see my cursor, but if you see here is nature's tunichrome structure, right? On the top center. So that's marine organism. And you can see here the muscle, right? Marine muscle. I think this one you might have, many of you have seen it. Is also a marine animal. So that stick on the rock or rock in a seashore. So these animals have a unique protein in their structure, which is DOPA. So if you see the chemical structure of DOPA is on the bottom left, then you can see it's a phenyl ring with 2 hydroxyl group. So this is a catechol group. So when these marine animals have these DOPA protein in it, that catechol group is basically oxidized and it act as adhesive in so because the sea is a wet environment, right? So even though in wet environment it stick, these animals stick to the rock or any like a hard surfaces. So we have mimicked this strategy. We have incorporated these dopa like moieties, that is basically pyrogalol or catechol, onto the chitosan, because I have already mentioned the advantages of chitosan or chitin. So that's why we have used chitosan as a main substrate, and then conjugate this dopa moiety onto the chitosan, and then we develop these tissue adhesives. So this is the synthesis process on that how we have incorporated the catechol group. So we have used gallic acid as a catecholic moiety and then conjugate on chitosan. So as I mentioned, the chitosan has amine group because it's a deacetylated chitin. And we use this EDC and NHS as a like catalyst and co-catalyst that basically use the carboxylic group of gallic acid, convert it into the NHS ester group, and then bind on the amine group of chitosan. So this is basically amidation reaction to bind gallic acid onto the chitosan. So now we have this moiety like chitosan gallic acid. So the chitosan unit has a moiety that has catechol group, right? So this moiety, as I mentioned, has a ability to stick in wet environment by the oxidation process. And so this is the characterization, like we have characterized it using FTIR and NMR spectroscopy to confirm that the conjugation has happened and it's a chemical conjugation, it's not a physical mixture. And once the material is developed, then we did the test. So this is the test to check the how strong is this surgical glue or adhesive. We need to check in wet environment, right? Or similar to we need to manage a system similar to our body. So we have used this. If you see the left top side, you can see aluminum bars and then porcine skin. Porcine is a pig actually. So we use a pig skin as a test, because that is how we do like a ex vitro and ex vitro test on a person's skin, because it's a biological material. And so we use this person's skin, we fix it on the aluminum bars, and then we apply our tissue adhesive that we have developed, and then allows it to cross link. We have used two different strategies, like for because this catechol actually forms complex with any metal ions. So if we add ferric anions, then it forms complex, and basically it acts as an adhesive. And also it's like a covalent complex. So if you anyway oxidize it, like periodate oxidation or something like that, then it also cures. So that's how it functions. So we have applied this material, allow it to cross link, and then we pull these two aluminum bar in different direction. So that's how mechanical testing works. So it's this a technique is a single lap shear test. So lap shear is like pulling two bars on the opposite side. So we applied known load and we can see, okay, how strong is this hydrogel? And based on that, we can know like, okay, like what is the known load on this test. So here is the column chart that shows a result, right? Like a different columns. So you can see the fibrin glue. Fibrin glue is a commercially available glue that is used in hospitals in surgical procedures. And this one is the one that we have fabricated is a CSGA, it's a chitosan gallic acid basically, and then using different cross-linking strategies. So yeah, it shows a very interesting adhesion strength, means it is strong enough, and it is actually in fact better than the commercially available one. Then we have also electrospoon it, so which means we have a hydrogel, but we want to make it in a solid form, in a fibrous form. So we have used this electrospinning techniques. So that is actually how it works. So we apply high voltage, and you can see this blue one is a I don't know, can you see my cursor? Can you see my cursor? No. Here, can you see my cursor moving there? Anyone from audience?
No, sir.
Yes,
Okay.
sir.
Okay, you can't see my cursor. Okay, so if you see the top middle figure, that is basically you see the blue syringe that has like electrospoon fibers, and you can see the CSGA solution. So we applied voltage difference in that, and then due to that electrostatic difference, that materials like a hydrogel in the syringe that basically drag into the collector, and that forms nanofibers. So we have fabricated these nanofibrous mat that is basically used as a hemostasis function. So
Can you use pointer, please?
pointer, okay,
Try to see somewhere.
okay, okay, is it fine now?
Yeah, now it's good. Yeah.
Okay,
Now
okay,
more
great,
visible.
yeah, great, thanks, yeah. So yeah, it's much better now. Okay, so this is how electrospinning works. So we have used this material for two different strategies. One is tissue adhesives, and the second one is to produce nanofibrous mat. We also measure the fiber diameters, like how thick are the fibers. So it's in the nano range. Yeah, so this is the tissue adhesive uh, project. Another interesting research I have carried out is on guided tissue regeneration. So guided tissue regeneration is a widely employed dental procedure using a barrier membrane. This is the barrier membrane to reconstruct the bond defects caused by trauma or disease, or to protect the implants. So for example, we have these dental implants, and we have this like a disease or trauma, then we need to protect these by applying this membrane. Otherwise, the implant will not have a mechanical strength, right? So this is the guided tissue regeneration membrane. So this membrane has normally two functions. One is so this is the teeth with a disease or a defect. So this is the teeth bond that basically supports. when it has a defect in it, we need to apply this membrane, and this membrane has two functions. One is to provide a barrier between here. It needs to provide a barrier here because this pink part, is a soft tissue. We call it gum, right? So this gum is actually soft tissues, but there is also this part which is bone. Basically, it's a hard part. So this soft tissue it grows very fast than the bone. So if it grows fast and if we don't have this membrane, then the soft tissue will infiltrate in this part and the bond will not form. So which means our teeth will have poor strength. So yeah, our teeth will be like wiggly in like a very short period of time. So that's where this guided tissue regeneration membrane is used. So yeah, this is the so this was the strategy to fabricate this membrane. So the membrane that I have developed has two phase. So it has two layers. Okay, it is called Janus because it has two phase, like a two different function, two different side, 1 and 2. So the first layer is composed of chitin, is as I mentioned previously, it is chitin, and the second layer, the second functional layer is composed of a kind of polymer that I have synthesized that I'm showing you. So the polymer is basically PMT in short. This one. This polymer is synthesized by random copolymerization of two polymers, MPC and TSMA. Both of these have a certain purpose to use. MPC is basically biomimetic approach. So it's basically mimic the cell membrane structure. This phosphorylcholine group is a zwitterionic group, which means it is very hydrophilic. So when we have hydrophilic surface, then it is, like the water contact angle will be very low. So is it attracts water, and when we have water, then the cells addition will be little bit less. So like for any cells to grow, there should be attachment. But since we have the water there, water barrier there, the attachment will be less. So that was the concept. We have used this MPC as an active ingredient, and then TSMA is anchoring group. So this methoxysilane group here can bind onto chitosan. So that was the purpose of using TSMA. So we have synthesized this random copolymer of these two monomers. So this is the final polymer. We have characterized it using FTIR and NMR, and then fabricate the membrane. So here is the membrane. As I said, it's a two layer. The first one is a PMT, is a synthetic one, and the next layer is a chitin membrane, right? Both have different function because this one will be used to create a water barrier so that the soft tissues cannot adhere. So for example, if we go back to this, yeah, the outer layer facing the tissue inhibit the soft tissue filtration because it has a water layer there. So that's how the barrier property works. And also the chitin is known for bone regeneration. So it actually support the osteogenic process, which means our body has a self-healing property. So it generates over the time. So that's how these chitin work. Yeah, so this membrane has two layer, and one is good for bond, one is good for gum. And we have done these in vitro cell studies, which confirms our hypothesis. Like one layer is good for soft tissue, one layer is good for hard tissues. We have used two different cell lines. For bone, it is osteoblast cells. So we use osteoblast cells from mouse, and for soft cells is a fibroblast. So we use fibroblast cells from mouse. and we do the cell viability test and cell proliferation, like to see how the cells are growing and how cells are happy. So yeah, this was the project for guided tissue regeneration membrane. So the next project is again on dental application. So is to develop a dentinal hypersensitivity treatment. Okay, hypersensitivity treatment. I think you might know that what is hypersensitivity. અને yeah, ગુજરાતીમાં જો સમજતા હોય તો આપણે જે દાંત કળે ને એને, okay, that is hypersensitivity. So the hypersensitivity is a sharp and unpleasant pain caused by exposed dentinal tubules when enamels outside of the teeth wears away. So this is the cross section of teeth. If you see here, normal versus um, sensitive teeth. So these normal teeth normally have like gum covered to the bone part, but when we have like when we have disease or when we have a poor teeth health, then this gum is little bit like swollen, and that's where the space is visible, right? And when we eat hot or cold food, so if you see this picture, when we this gum is a little bit swollen, then these teeth, the dentin part of the teeth has a tubes. It's like a very hollow channels, like very small in the micron scale. It has a small tubes or channels that goes through the center of the teeth. So when we have this gum problem or gum health issue, then these tubes or mouth is open, right? And when we eat hot or cold food or liquid, then what happens is like there is a changes in the pressure here, right? If you eat cold water, then the pressure outside will be different than the inside. So that's where this fluid in the tube actually cause the pain, because there are nerves underneath the dentin. So that's how the hypersensitivity happens. To overcome this problem, we need to use we need to block these tubes, right? Because when yeah, we need to block these tubes. So the teeth is mainly is made of hydroxyapatite. It's a mineral. It's like a calcium and phosphate in a different ratio and some amount of biomolecules. So that is a hydroxyapatite. So if we use hydroxyapatite to block these tubes, then the hypersensitivity can be cured. So we have used again biomimetic strategies like a catechol group that binds with metal ions, and then for catechol group we have used this natural polymer, which is tannic acid. So tannic acid is means like anything you see brown in the tree is basically due to the tannic acid. So yeah, we have used tannic acid with metal oxide and metal ions, and that solution uh, we have treated the teeth actually, not the whole teeth. What we did is we sliced the teeth and took a disc of teeth. It's a like thin disc of a teeth. So if you see these photographs here is basically a scanning electron micrograph of a teeth. So A1 is actually open tubes. So which means when we have a hypersensitivity problem, then these tubes are open like these. And you see the channels, the cross section of the channels. So yeah, this is how it looks. So when we treat these teeth disc with a tannic metal ion solution that we have developed, then what happens is like it passes through the channels, and due to this catechol and metal oxide, it basically uh, provide a site to regenerate the hydroxyapatite in a faster way, in a very rapid way, in fact, actually, because within 4 minutes it blocks the tubes So if you see this B1, right? If you see it's partially blocked, inside is still not happening, but the top is partially blocked. But after 7 days, if you see the cross section like B3 and B4, so you can see it's almost fully blocked, right? So these needles like a stuff here is basically hydroxyapatite crystals. So that basically far that basically increase the speed of hydroxyapatite formation. So top one is control. So if we don't use this solution, then you see here A3 and A4, there is no mineralization. The channel is still open. But if you use this solution, then you can see it's blocked. So if we have this hole like a blocked, then there will be no flow of the fluid, right? So that's how okay. Yeah, so that's how it works. And also we have tested in another way also, apart from a scanning electron microscope. So we have used this equipment. We place our dentin disc that we have tubes in it, and we pass a liquid through it, right? And then we calculate like what are the percentage of blockage. So it's like a before treatment and after treatment. And for treatment, we have two different ways, like a 4 minutes and 7 days. So we have used this experimental condition. We have used different ions like titanium dioxide, SR2 plus, then iron, calcium, and like all those metal ions and oxides. And you can see here in the bar chart, the like titanium dioxide shows almost 70% of blockage in 7 days, and SR2 plus shows almost 80% in 7 days, so which is quite significant. And the another advantage of using this solution is so let's go back to the previous one. Sorry, my screen is frozen. Okay, yeah. So when we have these hypersensitivity problem, when we have these tubes open, then the internal part of the teeth has a yellow color than the outside. So if we have these tubes open, means our teeth are yellowish than the healthy teeth. So, but when we use this strategy to block the tubes, then the yellow part is blocked, right? So your teeth, the color of your teeth will also change. So we have done like two tests. One is color change and the one is whiteness index. So in color change, yeah, in color change, you can see this is the line, the horizontal line shows the minimum requirement of color change. So you can see like titanium dioxide and silicon dioxide shows is a way higher than the minimum required um, guideline. Yeah, and when in terms of whiteness index, the titanium dioxide shows so very promising results, because titanium is also known for its whitening property. So this solution has a two advantage. One is to treat the hypersensitivity, and one second one is to look better. So it's like aesthetic properties as well. Uh, yeah, so that was the hypersensitivity and dental applications. The next one is cellulose, is about cellulose. So we have developed this hydrogel using some chemistry. So you can see here is a structure of cellulose, as I shown before. It's a 1,4-beta linked glucose, and it has a CH2OH group, and other are like a secondary hydroxyl group. We have used this primary hydroxyl group CH2OH, and we modify it to the carboxylic group using this chemistry. So we have used tempo reagent, and we have used sodium bromide and sodium hypochlorite as a catalyst. And using this chemistry, we have oxidized these only, it's a selective oxidation because it only converts secondary hydroxyl group, primary hydroxyl group to the carboxylic group, not the primary one. So it's a selective oxidation. After the oxidation, we have given some mechanical treatment. So it's basically shearing the fibers, and that basically produce clear transparent hydrogen. So if you see papers here, so this is the start material, like a chunks of paper here. This was the starting material. After the oxidation and mechanical treatment, it turns into the transparent hydrogel form. This hydrogel has a very unique property. It's a it shows shear thinning property. So what is shear thinning property? If we shake this hydrogel, that it turns liquid. But when you keep on rest for some time, then it again go back to the gel state. So that is the shear thinning property. We have explored those properties using rheology. So rheology is nothing, it's just like study of how material is flowing, uh, what is his viscosity, and what is its like a modulus. So that is the rheology. So we have explored the rheological properties using different shears or a different like strain on it, like a shear, sonication. We have also used pH changes, also use cross-linking strategies by applying ions. So that's how we explored the rheological properties. And in all four cases, it shows that when we apply the external stimuli like a stress, pH, sonication, or cross-linking, it goes to the liquid state, and on resting, it come back to the hydrogel state. So you can see here, it's a liquid form, but when you rest it, it goes to the gel state. So what is the use of this type of hydrogel? This is to use for injectable hydrogels, because for cell therapy or for like tissue engineering, if we want to inject cells in our body, or if we want to do the cellular study, then we need some matrix, right? So this hydrogel can be used as a good matrix because we can make it liquid, just add cells, and we can achieve the cell suspension. But when we rest it becomes solid, so we can use it as a cell matrix for any cell studies as a model. So that's what its application. And the another application is 3D printing. So for this study we have used cancer cells, and then stem cells, yeah, those type of cells. And for 3D printing, I will go more into detail because it's very interesting, and maybe it's very, some of you might have not heard about it. So this is about 3D printing or 3D bioprinting with cellulose hydrogels. So 3D printing is basically a layer by layer deposition of materials, but in a very confined way. So if we have this 3D CAD model, for example, this is a statue or something, then what happens is like we can print in a 3 dimension like this using our materials. But how? So what do is we first make this CAD model is using AutoCAD or like any CAD software. We use this 3D model, and then we use software that basically slice this model, so in 2 dimension. So basically it's like a 10 100 or thousands of layer um, vertically that is combined. So basically each layer is 2 dimensional, but when you stack them together, it make 3 dimensional object. So it produce layer by layers, and then it generates tool path. So tool path is basically how this, suppose we have hydrogel in this syringe, then it deposit in a certain way that it makes these physical objects. So this is very useful, and it's a very interesting topic recently in tissue engineering, because of its we can create very complex structures and geometries. So that's why it's very interesting. This was my project on the 3D printing. So this is the 3D printer. It has 4 component. So let's start with the first. This first is a syringe. So we can load our hydrogel or any extruding material here in a syringe. This tube is basically is connected to pump. So it applies pressure in the syringe. Due to the pressure, this material inside the syringe is extruded on these 4. This is a platform, basically stays. So this head moves X and Y, and this stays moves Z direction. So basically it print in that way. So using these, yeah, and there is a needle here. Third one is a needle. The tip of syringe is a needle. It's basically, we call it injection, right? So yeah. Um, Using these 3D printing technology, I have started from 2 dimension. It's basically grids, like XY grids, to some like spider webs and like vascularization kind of structures, to the miniature model of these, like a heart, brain, octopus. So yeah, I just made this 3D model in the computer, and then we can give command to this printer, and it print actually layer by layer. So okay, I will show you this video. If you can see, okay, can you see this? Ah, it's not playing. I don't know. Wait. Okay, can you see the video playing now?
Yeah, yeah, noise playing.
Okay,
Yeah,
yeah.
yeah.
Okay, so this is how it works. So you can see here the head is moving X and Y. The pink one is basically tip or a needle, and this white one is a material. So yeah, it moves like that. So you can so you can fix the tool path using softwares, and that basically prints whatever you want.
It is computer monitor, is not it?
Sorry.
Is computer monitor is?
Yeah, it's all by software, but we can choose the design, we can draw the design, and then we can process with it.
Yeah,
Yeah.
so you have to give the design to the system, and then accordingly it will print on it, right?
Yeah, so first we make these sorry. Okay, yeah, first we make this CAD design. It's like any 3D design in the computer, like AutoCAD, right? And then we use slicing software that basically generate a tool path. Is basically a motion of this head. So the slicer tooth generate a motion, like how these head should move in each layer. So that basically make the 3D design. Yeah. So we can, how useful is it? So we can generate a miniature model of any, like a tissue. We can mimic the tissue models, because all tissues have different properties, and sometimes we need like a very specific shape of geometries, like a cylinder or a sphere or those kind of things. So that's where this 3D printing is very useful. Even recently there has been like very complex printing, like cartilage, bond, those kind of printing as well. I will show you the some of my example in next slides. So this is not about tissue engineering. It's a quite different from the title of this talk, but still I would like to show it because it's a 3D printing, but it's in large scale. So you can see here, it's a robot, right? It's a robotic arm. It's called robotic arm. And we have used this technology to generate 3D printed chitin or cellulose. So yeah, this project is about large scale 3D printing of biopolymers. This one has a environmental application. So we have used biomimicry again, like using these fungus. So this fungus has 2 components, cellulose and chitin. we mimic the composition and develop this material that is like a white, you can see white, sorry, white material. Yeah, so we develop this material. It's very cheap, it's very good to print, and using this material and technology, we have initially printed 1.3 meter long wind turbine blade. So it's like આપણે પવન ચક્કી જેને કહે ને, પવન ચક્કીનું પાંખ્યું, હા. So in starting, it was 1.2 meter long, just to show the proof of concept of this technology and material. And then later on, we have printed this 5 meter long structure using this technology and material, and which is the biggest biological structure ever printed till the date. So that's the record. Yeah. So this is a very interesting material. It can act as an adhesive. This wind turbine is 3D printed, is coated with the same material. This material act as a glue. So it's very lightweight. It's very cheap. This material is even cheaper than plastic. It's like maybe $2 per kilogram. So it's very cheap actually, and it's sustainable because it use natural sources. So its use is basically a substitute of plastic because it is biodegradable. You can use this material, and once you are done with the application of it, you can just throw the material, and it's all consumed by nature. The second one is again is very similar, but it's circular manufacturing and bioconversation. So what happens is, like, even though there are like trees and the sea animals everywhere, but some of the region might not have the resources for example, chitin. Many countries doesn't have the sea, right? So that's how this circular economy and manufacturing works. So we use this fly, is a black soldier fly. It's a bee actually. આપણે જેને માખી કહીએ ને. So we use this black soldier fly as a source of chitin. We extract chitin from it, and on the another hand, for cellulosic waste, we use a waste of paper. We again made material which is printable, which is used as a bioplastic, and then printed this 5 meter strong structure. So the advantage of this material is basically circularity. So let's say we have uh, okay, we have this waste, like okay, I forgot to mention that why we have used black soldier fly, because the black soldier fly has a very rapid life cycle. It becomes adult within 14 days, and it has a very high amount of chitin, and we don't need to kill them, because when there is a life cycle of this bee. So when it becomes, so initially it is egg, and when it becomes a pupa and then adult, then this shell of the bee is basically a source of chitin. I think આપણે બધાએ પેલું સાપ આવે ને, સાપની કાસળી ને, yeah, similar like that. So it's basically a shell of pupa. So we use that. So it's again, it's a waste. We use paper waste as a cellulose source, combine both the ingredient and then develop this material, and this is substitute of plastic. So we can 3D print it in a large scale. Even we can produce furniture, sports, any like architect stuff. And then when we are done using those type of like components, we can just throw it. So it's again, it's a biodegradable, right? All composition is natural, so it again go back to the nature. So it's a fully circularity um, of this manufacturing and supply chain. So that's how it was the interesting. Yeah, that's, I think, more of about my research. And I would like to thank our team and yeah, guides, colleagues, collaborators, and sponsors for these supporting this work. And yeah, thank you for your attention. And Yeah, I think it's quite complex. Some of parts are quite complex for you to understand, but I can like explain you in a very simple way if any of you are interested. Yeah. So if you have any questions or comments, yeah, please ask.
Jamin sir, Bhagani sir. Ashit sir. Hello. Might be Ashit sir. So by the time students, you are free to ask questions. If you raise your hand, okay, sir will take your call. Okay.
Yeah, any question, like you can ask
Yes,
even
yes.
like a very
Any
simple question.
question that,
Yeah.
yeah, it's good to ask question. So that's a good practice, as you know. So because that is what this kind of activity going on.
Yeah.
Not see how the 3D printing, you might be heard many places, but you have some real feeling, even that being used for the making the human body parts. So is not it a great things to think about it, right? Huh? Okay. Um, yeah, Naresh, it's a very great talk, I would say.
Thank you
Uh,
so much. Yeah.
yeah, because I know 3D printing actually. I was using in University of Alberta.
Oh, that's great. Yeah.
So that is because to making some kind of small piece for instrument, some kind of valve. Yeah. Now I see you are using the technology to in medicinal field. Okay.
Yeah, there
And
are like several types of 3D printers and
3D printers.
yeah.
Yes. So that's a great one. I think Jaimin is here. Jaimin?
Yes, yes.
Ashit sir also there. Okay, good. Uh, yeah, there, sir, just take some questions.
Yeah.
Ashit sir,
Yes, sir.
could you see some questions from students?
I
I want
think
to,
that
yeah.
one student Shivam has a question.
Yeah.
Yes, researchers in which they have imparted preservatives and antimicrobial agents in PLA. Is it same for this hydrogel also?
Yeah. So PLA is a little bit, so printing PLA is a little bit different than printing hydrogel because PLA how it works. So in case of PLA, you have these what do you call filaments, right? So you have this that PLA filaments, you heat it, and then when it deposit on the bed, it actually cures. That's how PLA works. PLA is not fully natural polymer because it needs modification uh, to use, right? It's not ready-made natural polymers. Another thing, the second thing is biodegradability. Even though like many of the researchers claim that it is biodegradable, but it requires certain conditions or environment to degrade it. So that's how it is different from the topic that I covered. So cellulose and chitin is readily available. We can use directly, and it can degrade in a natural condition. So, yeah, for the preservative and antimicrobial agents, yeah, there are like so many ways to get the antimicrobial properties of any polymers and biopolymers. Even like some metal particles works as a antimicrobial. So yeah, that works on these as well. Yeah, hope that helps.
Yeah.
Yes. So any other student have query, question?
Student, faculty, anybody, please raise your hand. We will allow.
Yeah, no worries. You can ask any
Yeah.
question. It's
Yeah.
more like a interactive one, so
Yes.
don't
Yes, yes.
hesitate. Yeah.
Yes. Take this opportunity and be interactive. There is Professor Ali actually just thanks you. Just to
Thank you
make
so much,
this technology.
Professor
Yeah.
Ali. Yeah, thank you so much.
Yeah. Ali actually from Egypt, you know.
Oh,
He's
that's great. Yeah,
the one of the outside participant. So
that's
thank
great.
you,
Yeah.
Professor Ali. So joining us also. and shows your interest. I don't know how you got this information, but anyway, it is pleasure to have you in our this meeting. Okay, so please
Yeah.
be tomorrow also, Professor Ali. Okay, and student, raise your hand, put up your question. Hashit sir will read your question or allow you to speak to Naresh, Professor Naresh. Okay. So, yeah. So far, no more question. I think, yeah, what I think, yeah. So, Hashit sir, yeah.
Yes.
If,
Okay.
yeah.
So if there are no any questions, then I would like to thank Dr. Naresh. He's a very good friend of mine,
Yeah.
for giving his time and his very precious time to us.
Glad to be here. Yeah, glad to be here.
Student,
Yeah.
yes.
somebody has raised the questions. I think
Yeah.
raise hand. Mustaq Junkia. Can you allow? I think.
Yeah, sure. I
Yeah,
can't see the question.
yeah, please.
No, he has not written in chat box.
No,
Okay.
raise hand. Maybe Naresh has a permission to allow him and speak.
Yes, Professor Ali, we will give you the recording link, and from that you can download it.
Yeah.
So I hope that students might have learned that these carbohydrate molecules are very small, but you can find that the enormous applications of it in day to
Yeah.
day. So it's a very basic chemistry, but that is very much informative lecture. And I once again thank Dr. Naresh for giving us this opportunity to share his research.
Thank you for having me. Thank you so much.
Nihal,
Yes,
Yeah.
Nihal, can
yes,
you,
yes, yes.
Nihal or Jamin, can you?
Thank Dr. Keyur Bhatt sir and our Dean of Science for hosting this series, this webinar. So from this Department of Chemistry,
Naresh sir, if you could stop sharing your screen, please.
Oh, so sure. Yeah.
we want to share you the gratitude.
Okay.
Is it good?
Yeah.
Yes, from Department of Chemistry, we would like to share you the statement of gratitude.
Thank you so much. Yeah.
Sanadia.
Yeah.
We will mail you this certificate.
Thank you.
And also there is one session tomorrow that will be given at also at 1 p.m. So thank you all of you for staying with us, and thank you.
Students need to stay back. They have a feedback form has to fill up.
Yes.
A minute. Students, you should not leave right now. Stay back for a few minutes. And Naresh sir, if you would like to leave, it's
Yeah.
okay. Otherwise, you can stay back also.
Yeah. Thank
Just
you,
take the
Dr.
feedback.
Das and
Yeah, thank you
Dr.
very much.
Hasid. Yeah. Thank
Yeah.
you. Bye.
Yeah. Thank you, bye. Yeah.
Yeah.
Yeah. Jamin, can you please post the feedback
Yes,
link?
sir, students, I will be sharing you the attendance link that will be open for next 10 minutes.
Feel free to put your suggestion. Okay, students. And tomorrow you must have joined in time. And also we have make a arrangement, good arrangement over here. So you be in the campus also. That we more like to be here. Okay.
Okay, students, so all the participants are requested to fill out the attendance link that has been shared in the chat box. Keep in mind the chat box, the screen will be open for next 10 minutes only. So you are requested to fill it out as early as possible. Students, you don't need to write your enrollment number in the chat box. I have shared the attendance and feedback link. So please fill that out.
So your feedback itself count as attendance. Okay, don't worry about it. So just fill up your things and okay. So wait for a few minutes, Jamin, and then we can close the session.
Yeah.
And
And
might be we
tomorrow,
can, yeah.
student,
Announce it,
tomorrow
yeah.
you have to join in the same link at 1 p.m.
Okay. Nihal, might be you want to stop the recording, is it? Nihal?
Sir, you have to stop the recording. You are the host.
Oh,
Once again, don't write your